Hello and welcome everyone. In last class, we learned about DNA demethylation. So methylation marks, which were initially thought to be permanent, after the discovery of 5-hydroxymethyl cytosine, it was established that those DNA methylation marks are not permanent, rather they are dynamic, and the erasure of this methylation marks results in DNA demethylation. So today we'll be learning about possible mechanisms by which DNA demethylation occurs. Possible mechanisms by which DNA demethylation occurs. Okay, so in the DNA methylation, what happens? The CH3 group is added on the cytosine ring of CpG dinucleotide with the help of DNMTs. We have learnt about this till now. Okay, so in the same way, the one possible mechanism for DNA demethylation could be direct removal of CH3 group of 5-methyl cytosine. Okay, so like when the when on the cytosine CH3 group is added, it gets converted to 5-methyl cytosine because this CH3 group is added on the 5th carbon of cytosine. So we can directly remove this CH3 group and convert it back to cytosine, right? But this is not possible. Why? Because formation of carbon-carbon bond is feasible, but breakage of carbon-carbon bond, which is highly stable, this is not favorable. This is thermodynamically not favorable, and that is why direct removal of CH3 group is not possible. We don't know yet whether this happens or not, but this is thermodynamically least favorable. This event, direct removal of CH3 group, is thermodynamically least favorable, and that's why we say that this is not possible. So the other mechanism for DNA demethylation is base excision repair pathway. So how does DNA? So base excision repair means you replace a misplaced base. Okay, base excision repair. This is the DNA repair event. And when this DNA repair event occurs, when there is some disruption in the DNA sequences, when the DNA sequence is not as desired, and that's when DNA repair occurs. So when we were learning about DNA methylation, we learned that 5-methyl cytosine can undergo deamination, and it gets converted to thymine. Okay, so in the sequence, let's say this was the initial sequence. Let's not take a G here. Let's say this is the initial sequence. Okay, so this will be the double-stranded DNA, right? So this will be the sequence. So only this only on this cytosine CH3 will be added. Okay, now when this 5-methyl cytosine undergoes deamination reaction, it will be converted to thymine. So this the upper strand will somewhat look like this: A T T G C C A T. Okay, and the lower strand will be same: T A G C G G T A. Okay, so here you can see T and G, this mismatch is created. And now to correct this mismatch, DNA repair machinery plays its role. Okay, so on this thymine, the thymine which was created by deamination of 5-methyl cytosine, an enzyme which is known as TDG. This enzyme is thymine DNA glycosylase. Okay, so this TDG enzyme is responsible for removal of thymine. So as soon as this thymine is removed, so what happens is first step is deamination, then second step is TDG mediated removal of thymine, and then third step will be addition of new cytosine. Okay, so this new cytosine will be unmethylated, and this is how this excision repair pathway gives rise to the demethylated DNA. So from 5-methyl cytosine, we got to the unmethylated cytosine. The other possible mechanism for DNA demethylation is NER pathway. So as BER was base excision repair, NER is nucleotide excision repair pathway. Though this pathway is a bit costly, means it costs more energy than base excision repair pathway, but this is also an possibility for DNA demethylation. So in the base excision repair pathway, we remove a base, while in nucleotide excision repair pathway, a whole patch of DNA is removed. Okay, so the whole patch of DNA, the whole region of DNA where the methylated cytosine lies, this whole patch is excised, and then new nucleotides are added over there. So in those nucleotides will be unmethylated cytosines, right? And this is how a nucleotide excision repair pathway works. First, the whole patch of DNA is removed, and then new nucleotides are added. This is how the methylation mark is removed. The fourth, in those pathways, in all of those pathways, the first pathway, direct removal, this was not possible. Okay, and base excision repair pathway and nucleotide excision repair pathway, those are DNA repair pathways. Okay, those require a DNA repair machine. The fourth pathway doesn't require anything else. Rather, it is a very recently studied thing, because this was discovered by scientists when they got to know about TET proteins. Okay, so TET stands for 10-11 translocation enzyme. So 10-11 translocation. So TET 10-11 translocation family of enzymes. This family of enzymes contain different TET proteins like TET1, TET2, TET3. Okay, so all of those proteins, those are the enzymes which demethylate DNA. So how does they do that? Because we already saw that breaking of carbon-carbon bond is not possible in the physiological systems, because it is thermodynamically not favorable. Okay, so this TET enzymes have rather very new approach. Those enzymes oxidize the 5-methyl cytosine. So 5-methyl cytosine gets oxidized. Okay, so just a second. So 5-methyl cytosine gets oxidized, the oxygen is converted into carbon dioxide, and this 5-methyl cytosine gets converted into 5-hydroxy-methyl cytosine. So this was when the scientists got to know about 5-hydroxymethyl cytosine. They got to know that methylation marks are dynamic, means they can be removed. And at the same time, they discovered the enzymes, those state proteins, and were the enzymes. This was the new kid on the block, which was discovered in the epigenetics world, which gave us a very new insight on the DNA methylation. So 5-methyl cytosine is oxidized into 5-hydroxymethyl cytosine. And the cofactor of this reaction, the cofactor for TET enzymes is alpha ketoglutarate, which in the process is converted into succinate. Okay, so this oxidation doesn't stop here. This could go further on. So this is not the final. This is not the final product. This is the rather a series of oxidation steps. So 5-hydroxymethyl cytosine can be further oxidized. It can be further oxidized into 5-formyl cytosine. 5-formyl cytosine. Okay, again the cofactor for the enzymatic reaction is alpha ketoglutarate, which get converted into succinate. Again, the 5-formyl cytosine can be converted into 5-carboxyl cytosine. 5-carboxyl cytosine. So the oxygen will be converted into carbon dioxide, and alpha ketoglutarate, the cofactor, gets converted into succinate. So this is how TET-mediated demethylation occurs. So this is it for today.